The Making of the Fittest, DNA and the Ultimate Forensic Record of Evolution by Sean B. Carroll. Narrated by Patrick Lawlor. Copyright 2006 by Sean B. Carroll. This unabridged audiobook is published by arrangement with Sean B. Carroll and W. W. Norton and was produced in the year 2006 by Tantor Media Incorporated, which holds the copyright thereto. This audiobook refers to several figures and tables which can be found on the last disc of this audiobook or at www.tantor.com, keyword Fittest. Preface. Beyond any reasonable doubt. Facts do not cease to exist because they are ignored. Aldous Huxley. In 1979, Diana Green, 9 months pregnant, was severely beaten, and her unborn child died from the trauma. Even though she had amnesia and could not spell her name at trial, she testified that her husband, Kevin Green, had inflicted her injuries. Green was convicted of murder and attempted murder. In 1996, workers at the California Department of Justice Laboratory processed the 17-year-old murder scene evidence for DNA analysis. By comparing the sample's DNA profile with those of Green and in a newly created offender database, they discovered that the original sample and samples from 4 other murders matched that of a different man, Gerald Parker, who was then in prison on a parole violation. Confronted with the DNA evidence, Parker confessed to the crimes and was subsequently sentenced to death, and Green was freed from prison after serving 16 years for a crime he did not commit. More accurate and rigorous than fiber or fingerprint analysis, and far more reliable than eyewitness testimony, DNA analysis can provide conclusive proof about who was or was not at the scene of a crime. The authority of DNA evidence, and many cases similar to Green's, have led to a revolution in the criminal justice system and a vast increase in the use of DNA testing to both convict the guilty and exonerate the innocent. Many crimes that would have been unsolvable in the past are now solved routinely, including cold cases several decades old. The number of exonerations is also growing. The Innocence Project, an organization that provides pro bono representation for DNA-based appeals, reports more than 150 exonerations over the past 13 years, including many individuals freed from death row. The power of DNA testing extends far beyond criminal justice. The determination of paternity is now definitive, and testing for carriers of genetic diseases is now routine thanks to DNA science. But there is one arena where that power is not yet widely appreciated, in what one might call the philosophical realm. Just as the sequence of each individual's DNA is unique, the sequence of each species' DNA is unique. Every evolutionary change between species, from physical form to digestive metabolism, is due to, and recorded in, changes in DNA. So too is the paternity of species. DNA contains, therefore, the ultimate forensic record of evolution. This presents an interesting irony. Juries and judges are relying on DNA evidence to determine the liberty or incarceration and life or death of thousands of individuals, and it appears that citizens in the United States are 100% supportive of this development. Yet in the court of public opinion, some 50% or more of the U.S. population still doubts or outright denies the reality of biological evolution. We are clearly more comfortable with DNA's applications than with its implications. Over a century ago, William Bateson began one of the first important books on evolution to appear after Darwin's time with the exhortation, "If facts of the old kind will not help, let us seek facts of a new kind. That the time has come for some new departure, most naturalists are now, I believe, beginning to recognize." With DNA science penetrating so many facets of everyday life, it is again time for a new departure and to seek facts of a new kind. My goal in this book is to present a body of new facts about evolution gathered from DNA evidence. Over the past few years, biology has gained unprecedented access to a vast amount of DNA evidence from all kinds of organisms, including humans and our closest relatives. In just 20 years, the amount of DNA sequences in our databases has grown 40,000-fold, with the vast majority of that coming in this new century. To put that number in perspective, in 1982, our total knowledge of DNA sequences from all living species amounted to fewer than 1 million characters. If printed onto pages like those you are reading, the amount of text would fit easily into one volume about the size of this book. If all of the DNA text that we now have was printed into volumes and stacked, they would reach more than double the height of the 110-story Sears Tower in Chicago. This library of life is growing by more than 30 stories per year. Inside these books is the raw DNA code for the making of all sorts of bacteria, fungi, plants, and animals. The decoding of these texts, composed of almost infinite permutations of sequences of just 4 letters, A, C, G, and T, now presents one of the greatest opportunities in the history of evolutionary biology. Biologists are mining this rich new resource to investigate and solve some of the most fascinating mysteries in natural history and to reveal, in unprecedented detail, how all sorts of important traits have evolved in nature. In this book, I will tell the story of how the new science of genomics, the comprehensive and most especially the comparative study of species DNA, is profoundly expanding our knowledge of the evolution of life. Genomics allows us to peer deeply into the evolutionary process. For more than a century after Darwin, natural selection was observable only at the level of whole organisms such as finches or moths as differences in their survival or reproduction. Now we can see how the fittest are made. DNA contains an entirely new and different kind of information than what Darwin could have imagined or hoped for, but which decisively confirms his picture of evolution. We can now identify specific changes in DNA that have enabled species to adapt to changing environments and to evolve new lifestyles. This new level of understanding provides more than just definitive forensic evidence. It includes some surprises that expand our picture of evolution. For example, in the DNA record of every species, we find fossil genes. These are bits of DNA text that were once intact and used by ancestors, but that have fallen into disuse and decay. These relics are an entirely new source of insights into traits and capabilities that have been abandoned as species evolved new lifestyles. The DNA record also reveals that evolution can and does repeat itself. Similar or identical adaptations have occurred by the same means in species as different as butterflies and humans. This is powerful evidence that, confronted with the same challenges or opportunities, the same solution can arise at entirely different times and places in life's history. This repetition overthrows the notion that if we rewound and replayed the history of life, all of the outcomes would be different. DNA evidence is also revolutionizing the study and understanding of human origins and early civilization. While the sequencing of the human genome has grabbed most of the headlines, it is the decoding of the genes and genomes of other primates and mammals that enables us to interpret the meaning of the human text. Our genes contain telltale clues to both how we are different and how we evolved to be so. Many genes bear the scars of natural selection, of the battles our ancestors fought with the germs that have plagued human civilization for millennia. I wrote this book with a variety of readers in mind. First, for those with a keen interest in natural history, I will roam the planet to show how many fascinating species have adapted to boiling hot springs, caves, jungles, lava formations, the deep ocean, and other remarkable places. There is grandeur in this new knowledge of how changing one or a few letters in simple code can dramatically change the form or physiology of complex organisms. Second, for students and teachers, I have focused on what I believe are the best examples that illustrate the key elements of the evolutionary process while reinforcing and expanding our awe for the amazing diversity and adaptability of life. Most of the stories I tell are not yet in textbooks, but many will become key chapters in evolutionary science. And third, for those trying to sift through the rhetoric and pseudoscience of evolution's opponents, I have provided some background to understand the tactics and arguments used to doubt and deny evolutionary science, and plenty of scientific evidence to vaporize those arguments. The new DNA evidence has a very important role beyond illuminating the process of evolution. It could be decisive in the ongoing struggle over the teaching of evolution in schools and the acceptance of evolution in society at large. It is beyond ironic to ask juries to rely on human genetic variation and DNA evidence in determining the life and liberty of suspects, but to neglect or to undermine the teaching of the basic principles upon which such evidence and all of biology is founded. The anti-evolution movement has relied on entirely false ideas about genetics, as well as about the evolutionary process. The body of new evidence I will describe in this book clinches the case for biological evolution as the basis for life's diversity, beyond any reasonable doubt. Chapter 1. Introduction. The Bloodless Fish of Bouvet Island. When we no longer look at an organic being as a savage looks at a ship, as at something wholly beyond his comprehension, when we regard every production of nature as one which has had a history, when we contemplate every complex structure and instinct as the summing up of many contrivances, each useful to the possessor, nearly in the same way as when we look at any great mechanical invention as the summing up of the labor, the experience, the reason, and even the blunders of numerous workmen, when we thus view each organic being, how far more interesting, I speak from experience, will the study of natural history become? Charles Darwin, On the Origin of Species, 1859. It may be the most remote place on earth. Tiny Bouvet Island is a lone speck in the vast South Atlantic, some 1,600 miles southwest of the Cape of Good Hope, Africa, and almost 3,000 miles east of Cape Horn, South America. The great Captain James Cook, commanding the HMS Resolution, tried to find it on his voyages through the Southern Ocean in the 1770s, but failed both times. Covered by an ice sheet several hundred feet thick that ends in sheer cliffs, which in turn drop to black volcanic beaches, and with an average temperature below freezing, it still doesn't get many visitors. Fortunately for both my story and natural history, the Norwegian research ship Norvegia made it to Bouvet Island in 1928, with the principal purpose of establishing a shelter and a cache of provisions for shipwrecked sailors. While on Bouvet, the ship's biologist, Ditlev Rustad, a zoology student, caught some very curious-looking fish. They looked like other fish in most respects. They had big eyes, large pectoral and tail fins, and a long protruding jaw full of teeth. But they were utterly pale, almost transparent. When examined more closely, Rustad noticed that what he called white crocodile fish had blood that was completely colorless. Johann Rudd, a fellow student, traveled to the Antarctic 2 years later on a factory whaling ship, Vikingen. He thought the crew was pulling his leg when one Flenser, a man who stripped the blubber and skin from the whale, said to him, "Do you know there are fishes here that have no blood?" Playing along, he replied, "Oh yes? Bring me some back with you." A good student of animal physiology, Rudd was perfectly sure that no such fish could exist, as textbooks stated firmly that all vertebrates, fish, amphibians, reptiles, birds, and mammals possess red cells in their blood that contain the respiratory pigment hemoglobin. This is as fundamental as, well, breathing oxygen. So when the Flenser and his friends returned from the day's efforts without any bloodless fish, Rudd dismissed the idea as shipboard lore. Rudd returned to Norway the following year and mentioned the tale to Rustad. Much to his surprise, Rustad told Rudd, "I have seen such a fish," and showed him the photographs he had taken on his expedition. Rudd heard nothing more about the bloodless fish for 20 years. Then another Norwegian biologist returned from an Antarctic expedition with white-blooded fish from a different location. His curiosity reawakened, Rudd began to ask other colleagues voyaging to the Antarctic to be on the lookout for...
Sample complete. Ready to continue?